All right, it is time to get started for another ACS DOC Virtual Symposium. My name's Carl Hansen. I'm a member of the DOC Exec Board. I'm here with Scott Begley, who is our part of our virtual symposium organizing crew, and Rio, the man who provides the smooth jazz tunes and does all the work behind us. He's part of Emory and also facilitates these. We have a really good. Oh, and then Steve's just joined us. Wow, Steve got out of his meeting, so it's good to have you here. We've got a great symposium for you guys as people are clicking on and joining up. We'll do some formal introductions, but we have Martin Eastgate from BMS and Sophie Rousseaux from University of Toronto. We'll do formal introductions in a second. We just want to do a few announcements to get things started. So if you can go to the next slide, Steve's going to talk to. We got our next symposium is actually pretty cool. So Scott, maybe you can walk through briefly what we're doing in November.
Thanks, Carl. Yeah, really excited about not only today's talks with Martin and Sophie, but for next month in November 17th, same bat channel, we're going to have a PUI focused edition of the ACS Virtual Symposium. We're going to feature three exciting talks from PUI universities. We have Professor Lalonde from Reed College, Professor Johnson from University of Richmond, and another Professor Johnson from Hope College in Michigan, all giving talks about what goes on in their labs in a PUI research lab. And we're really excited to have them join us and talk about another side of research that we don't normally see in these kinds of sessions. So really excited about having that. It's going to be a lot of fun. So
I love Rebecca's title. It's really ominous. Generating publishable results with undergraduate researchers. Like, I was an undergraduate researcher, and so I know the struggle is real. So it should be pretty. They all should be good, but I thought that was pretty cool. So love to see her secrets for success. So
absolutely.
Awesome.
So, and thanks to all three for joining in and doing this. It was took a little while to set up, but I appreciate them making the time. I know at a lot of the undergraduate schools, the schedules are really tight, especially during the day. So I appreciate them making the time to speak with us. So thank you all.
There's a lot of great work being done there, right? And certainly the future of chemistry happens there. So it's very cool.
Yeah, we're going to talk another big topic, this being October, is a time for
it's baseball, right? You want to talk about baseball,
no,
Scott?
I don't want to talk about baseball, not after last night. No, it's the Chem Nobel was announced last week, and so of course, congratulations, Professor List and Professor McMillan on their Nobel Prize for development of asymmetric organocatalysis. If you haven't been living under a rock for a week or so in chemistry. So congratulations to both of you, both professors and all of the students that have brought this to fruition. So wonderful job, everybody.
Yeah, it's really remarkable. Like when this happens, every October, it's kind of exciting when the Nobel chemistry, it's like the one time like the New York Times will have an article about chemistry, right? Like that's very positive. And so it's really exciting when that happens generally, but in particular when it's an organic chemistry, very organic chemistry focused. It doesn't, it's not every year that happens. So it's really kind of cool. And then I think the three of us, Scott, Steve, and I, we're getting together talking, it would be good to talk about this. And of course, I don't think any of us know Professor List as well, but we all know Dave, and had, who's a great guy, like very approachable guy too. I've known Dave for a long time. You guys have all interacted with him professionally, and because he has a lot of associations with the companies that we've worked with. So really nice to see. I was actually with him like a few days before he got this. I hadn't seen him in years, and then open up the paper. Not open up the paper. I opened up the paper on my phone, but to see him win the prize is really great, and really great for the field that we work in. It got me, like, I think we were like reflecting, like, how many, like, I think it started off a discussion of like, how many reactions, like, have you run an organocatalytic reaction? And then then I was like, well, how many reactions like that are Nobel Prize winning reactions? Like, have I ever run? And we were kind of discussing this. And I have this sort of, because I think I'm kind of, in my heart, like, have no social life. And so I have a lot of free time to, besides my kids and my family. But like, I had some time on Sunday, and I just started to try and like go through like, okay, how many Nobel Prizes are organic chemistry focused, or like, could you construe like, and I do go through the list like every year now and look through them and like, do a little bit of Googling. And so I put this list together. What's remarkable is the ones outside of this group. There's a lot that are arguably very like people who have influenced organic chemistry, or things that are just like so foundational for chemistry in general. But like, I put this short list together, and it was kind of an interesting process, like to go through it and see like what some of the contributions were. Like the stuff, like it's kind of like the old era on the left and the modern era on the right. So like things that resonate with me, having been a synthetic chemist for a while, like asymmetric catalysis. Obviously, that's what I got my PhD in. Metathesis, like that was like a phenomenon. Leading up to that prize. Cross coupling, organocatalysis. There's these sort of like lifetime achievement awards for like Woodward and Corey and their influence broadly. I like going back to these really old ones, because it never ceases to amaze me, like where the field was at that time. Like if you look at like the Grignard one is classic, right? Like that just fundamentally the idea that you could take a metal and an organic compound, react it, either to make an organometallic like Grignard did, or do a hydrogenation and do chemistry, like is so foundational. And it's just amazing to me, like if you think about like the tools and the equipment that folks had back then, like what they had to overcome to achieve the discoveries they did. I remember when I ran a Grignard reaction, I was like, fanatical about keeping it anhydrous and keeping it dry. It turns out like they're really not that reactive. Do you know what I mean? Like you can run them with like a little drying thing and stuff. So I just like, I think we were talking about this and wanted to share like our thoughts, or just like encourage anyone to have like go out and reflect and think about some things that from these past prizes.
Yeah, it's amazing how something like a Grignard, is so classic, and people. But how do people use them nowadays? They use them so differently now, with the sort of things like from that Professor Nochel or the late Professor Sneakus have done in the area of metallation and things like that. That it's completely used, traditionally, but also in many new ways. So like, how is some of this newer stuff on the right hand side going to be used? 20, 30, 50 years from now. It's amazing to think that, this is on the right, like you said, so foundational to what we do, and seeing where everything goes now, what's coming up next. It's amazing to think about. And,
and another gem is the elemental on that. Like there's so many gems, but like I would encourage people to, like, elemental analysis, like just imagine like that not existing, right? Like, and doing any work and stuff. But yeah, and then where has it come? Like where is that state of that now? Like just the concept of elemental analysis was novel back then. Cool. Like, okay, so we're gonna move on, but like I think what we're gonna do is turn it over. And as always, tweet us. I've been trying to like break up with my phone. Like I feel like I'm in an unhealthy relationship with my phone. So like I encourage anybody who wants to like get me excited. Like I'm ready to get excited about Twitter, but like I feel like it's against what I really should be doing with my phone.
Tell us what you think the next organic reaction on that list should be.
Yeah, love to hear that.
That'd be great. Yeah, what do you
or thoughts about any reflections anyone wants to share about Nobel Prizes, organic and otherwise, and other chemistries. Let's just keep it to chemistry.
Yeah.
And I promise to retweet it to all 5 people who follow me. That's a promise.
All
cool.
Righty. Well, let's get on with it. Thank you very much, Carl, Steve. So, and Rio, as always, thank you and CCHF for your support for this. So I'm going to introduce our first speaker today, Martin Eastgate, who is a director of process chemistry at Bristol Myers Squibb. And so Martin got his bachelor's from the University of Surrey in the UK and his PhD from the University of Cambridge under the direction of Stuart Warren, investigating sulfur chemistry, specifically thiouranium ions, which I actually had to look up. Sorry, dude. And the use in proline synthesis. And then after that, in a postdoc with Professor Denmark at the University of Illinois, he joined Bristol Myers and is currently an executive director and head of chemical research in chemical process development, and has a list of awards and outside activities too long to mention at this point because we're talking about Nobel Prizes for too long. But anyways, we're really excited to talk to Martin and hear his story, what he's been doing and went up to lately. So Martin, take it away. Thank you for joining us today. It's a real pleasure to have you.
All right, well, thanks Scott, Carl, Steve, and Rio for organizing the event and inviting me to talk to everyone today. It's great to be here virtually in your screens to meet up. It's amazing what we've been able to do managing against COVID, to connect in different ways, and really excited to be here today. Though I have to say, Scott and Carl, I think you guys need a podcast. That would be totally awesome. I'd tune in to hear you guys talk about chemistry and science any day of the week. So hopefully we can make that happen one day. Okay, so the stuff I'm going to talk about today is about innovation in the synthesis of pharmaceuticals, a little bit about what's going on in the pharmaceutical sector, and some elements about how we at Bristol Myers Squibb are starting to think about improving and continuing to develop novel science is going to help us deliver really important new medications to patients. Now, what I'm going to talk about today is not my work. It's not a story about me. It's a story about a really great and fantastic team, a really great community, and a really great set of collaborators that we have within Bristol Myers Squibb, many of whom are shown on the slide here. So I'm sure you can see some faces you all recognize, some new faces, some different people from across the MedChem team, the process group, and our academic collaborators within BMS. And it's really about the union of these mindsets and different perspectives that I want to highlight today as delivering on our promise of accelerating delivery and development and discovery of new medications through these great collaboration and through this great teamwork that the folks on this slide here have executed as we look in the process team to design and develop safe and sustainable routes to deliver high quality molecules to patients. So this is really everything I'm talking about is the output of this great group. So let's think about a little bit about what's happening in the evolution of pharmaceuticals. So the compound on the left-hand side here is Plavix, which is a very well-known compound developed to market in 1998 by Sophie and BMS. Really important anti-clotting agent. Millions of people have taken this compound, but as you can see, relatively simple in terms of its overall structure. Now, if you think about the last 20 years of drug development and the compounds that Bristol Myers Squibb specifically has put on market, these are the compounds that you'll see from our own portfolio. Starting move from atazanavir, which has a lot of naturally derived stereochemistry, and as you categorize from left to right, you'll see the molecules on the whole start to look a little bit bigger. They start to have more ring systems, generally more mass associated with them, and you start to see the installation of more novel stereocenters, maybe unnaturally derived stereochemistry, things like cyclopropanes and more complex architectures. And for all intents and purposes, one would think that these molecules are starting to get a little bit more complex. Now, we all also know that these molecules were discovered or some 10 years before they hit market. So if you want to look at what we're working on today, you really need to look at our more recent publications. And if you do that, you'll see compounds such as this really densely functionalized BTK inhibitor, which features two axial stereocenters embedded in a structure, really difficult molecule to think about making on 100 kilo scale. You'll see compounds like this FXR agonist from the labs of Dean Wacker at BMS, which contains, as you can see, the spirocyclic cyclobutene. Or molecules like malavaxan, which is a really great third generation now anti-clotting agent, which we're developing in collaboration with Janssen. And if you cast your eye from Plavix to this compound, which is Eliquis or apixaban, over to malavaxan, you can really see an evolution in the anti-clotting area and getting into some really beautifully large macrocyclic compounds. You'll see degrader compounds like 92480, or relatively simple compounds like mavacamten, or even things like drucosatinib, which is our latest NDA filing, which we hope to be influential for psoriasis patients. As you can note here, our first deuterated drug that's coming out of Bristol Myers Squibb. Really exciting diversity in the structures that you see before you. Now, you may think that it's just BMS's portfolio that has gone a little crazy with complexity, but I would advocate not. It's really an industry-wide phenomenon. A few years ago, Jun Lee in our group developed a really innovative computational method to assess molecular complexity that took the structure of molecules within the context of our ability to make them and looked at the evolution of virology drugs over the last 30 years. And certainly using that algorithm, one can see an increasing trend of complexity. And if you look out in the literature, you'll see many of our peer companies and partners across the industry talking about molecular complexity in their own environments. And certainly we have participated in the literature talking about some of the things that we've done within Bristol Myers Squibb. Now, you do have to continue to ask the question over time of, is it really complexity that's changing? And I think this is an important question of self-reflection as someone who talked about this topic a lot. So if you continue to look at all the NDAs that start to come out, you look at 2019 or 2020's diversity across the different kinds of molecules that have hit the market, or you just look at the structures of the compounds, either the ones that BMS has participated on in blue here, or more generally across the portfolio, you see a big diversity in structures from complex novel small molecules to kind of natural product like peptides and proteins, which are macrocyclic, to natural product analogs over on the right-hand side here, or even simple small molecules, which look a lot like Plavix. As you continue to explore, you'll see diverse modalities, things like oligonucleotides, or even peptide drug conjugates, which are taking complex cytotoxins linking to peptides, or more traditional antibody drug conjugates. And if you step back and think about what this means as an organic chemist in the pharmaceutical sector, I would say what an inspiring time to be a pharmaceutical scientist. We as organic chemistry are touching not just traditional like small molecules, which have their own wonderful complexity, really diverse and complex small molecules that are larger and more novel in their structure, but also getting into the worlds of bioconjugation and large biomolecule like chemistry. And to me, this is really inspiring for where we are as an organic chemistry community. Okay, so this is the topic we're going to ruminate on. And normally in these talks, I would ruminate on some of the portfolio project work that we've done, as well as some of the capabilities and data developments that we have going on in the organization. As I only have 25 minutes or so today, we're going to focus a lot on the capabilities and data analytics side versus the synthetic innovation. If you're interested in some of the molecules we work on, you can certainly see a lot of the references there in the literature that describe some of our innovative science towards specific projects. But we're going to focus on new capabilities and a little bit about data as well. So I wanted to start with data because I think it's foundational to how we need to move forward as a community. The ability to make decisions in highly complex environments and make great steps in our knowledge base really are going to require us to think more holistically about the data we have available. So let's think about what that means from a process chemistry team. So if you take this molecule, which is our BTK inhibitor I showed you earlier, this really dense axonally functionalized molecule, you'll see this a couple of times today, and you look at the kinds of experiments we ran on this project, just what I would call the technology-enabled experiments, the ones using our automated high-throughput experimentation platforms. We ran over 28,000 experiments just through HTE-like manifolds to come up with a commercially viable approach to that molecule. Not 28,000 data points, but 28,000 experiments. It's important to know these are highly curated experimental data sets, right? They're running an automated platform that takes down a lot of the natural variability of human operations. And this data set also contains both positive and negative hit information, right? Where positive reactions were and where reactions failed. And so the aggregate of this information, we felt, could really provide us a lot of insight that would help us with fundamental things that we deliver as a process chemistry team: route design, experimental planning, and how to think about sustainability. So we've published quite a lot on how we thought about leveraging this kind of data in sustainability derived places. So for example, as you think about a complex molecule, you could advocate this all-cis triaminocyclohexane here is relatively complex, but there's certainly hundreds of ways to make this molecule. We published on a couple, which are from cyclohexadiene or this monoprotected bisphenone. But as molecules get more complex, the number of ways we have to make those molecules expands exponentially, right? The number of options and decisions we had. So what we did was we took a lot of our efficiency and sustainability data, asked the GCI Pharma Roundtable members of Partnerships for Green and Sustainable Chemistry, took all the on-scale reaction data, and then developed an algorithm that could take some very simple information about the kinds of reactions you want or may want to string together in a proposed synthesis, and enable us to predict the likely sustainability outcome of a proposed synthesis without ever having to do the work. And this allows us to compare routes from a sustainability perspective without ever having to do the route scouting. So we can choose where to focus our effort for the biggest sustainable impact. We've taken our reaction screening data from catalysis and used molecular fingerprint approaches to almost look at docking between ligand and catalyst structure and substrates. So start seeing if we could predict which ligands are going to work in certain kinds of metal catalyzed transformations and be able to link that knowledge to the efficiency of the ligand synthesis itself to be able to say, is that ligand the right solution for that sort of problem, i.e. are we over titrating complex ligands with simple coupling chemistries, for example. And so we've been able to develop this predictive algorithm to take molecules from the literature, like this compound from the GSK portfolio, where they compared two different routes to make the key CN bond in the middle there, and predict which ligands worked for those two different scenarios. And actually the BMS data was able to predict which ligands worked for the GSK team, which I think was quite an impressive thing to do. More recently, Jay Stevens has been collaborating with Abby Doyle's team to deliver new ways to think about reaction optimization, developing what's called EDBO, which is experimental design by Bayesian optimization, a way to take highly complex multi-parameter design space and use Bayesian statistical design to decide where to look in that design space to run that next experiment to rapidly optimize the chemistry, and have developed quite an amazing platform to be able to take things like CNCO or CH arylation chemistries or things like Mitsunobu and very, very quickly get to some very, very optimized conditions. And that collaboration between Abby's team and the team at BMS has developed what I think is going to be a really great approach for us to optimize reactions quickly moving forward. Okay, so that's a little bit about how we've thought about data, and I think that's really an important area for us to continue to mine as a field as we move forward. The rest of the talk, I want to focus a little bit on how we're building capabilities within BMS. And, we can focus a lot on the kind of scientific or lab-based capabilities we have, but the ability to support innovation for the future is more than just our tools and the science that we have available. It's really a story, a question about people. It's about our community of BMS. It's about our philosophy of how we approach science, our culture of doing chemistry, and making sure we're bringing together the most diverse team we can to bring different thought perspectives into the equation. And I'm very proud to work for BMS as a company because the company really supports diversity, equity, and inclusion across our enterprise. So we have this fantastic people and business resource groups which connect horizontally across the whole BMS enterprise, diverse communities to help advance diversity within BMS. Over 11,000 members of our global team are now involved in these PPRGs, from our Bristol Myers Squibb Network for Women to our Pride Alliance to our Black Organization for Leadership and Development called BOLD, or many of the other elements that you see there. It really is an inspiring place to connect and collaborate and learn from each other horizontally outside of our own STEM fields. Within the chemistry community, Rebecca Green and Emily Cherney have developed a grassroots effort for outreach into different populations externally to bring women from the science community into BMS to look at what it would be like to explore careers in the STEM environment. So undergraduates and graduate students come in every year to learn what life is like in BMS as an enterprise, and to help them make career decisions for themselves as they transition from undergraduate to graduate and from graduate to careers in STEM. They've connected to our engineering team and our teams in drug product development to talk about outreach horizontally across the organization, and published this wonderful paper in J Chemical Education about our outreach efforts, which I think is a really influential paper and really important one for folks to read. It's also been really important for us to create allies within our community for advancing diversity goals. It really is so important to our future as an organization. And we've had important dialogues across our team on this topic. One recently that we conducted, which I thought was really influential, was around the Picture of Scientist documentary. Now, if you haven't seen this, I think personally it's a really, really important documentary. It details the challenges faced by women and minority populations within the STEM community, and to me is a really important call for action for us as a global community to simply do better to advance diversity within our field. If you haven't seen this and haven't watched it, I really encourage everyone on the line here to watch this important documentary and engage in this conversation with people around you. So that's been a foundationally important thing for us to action on, and things that I hope we are making progress against. Another topic that we've been working on is to improve our lab environment and to think about how we want to be executing chemistry in the lab in the next 5 to 10 years. And Rebecca, shown on the screen here, has been leading this effort for our next generation lab initiative and bringing really innovative things to think about how we want the lab environment to work for us differently, to amass the data we need to drive good decision making in the future, and has developed this TED-like talk, which is now up on YouTube. So if you're interested in our activities in Lab of the Future, please feel free to take a look and capture this UR code. Okay, so those are some really important foundational principles in terms of innovating for the future. It's also important that we pull from our past. So you'll see the reflections of this moving forward in the rest of the talk, but one of our early really important lessons in homogeneous metal catalyzed transformations, as we looked at scaling up these kind of complex transformations, was through the simple borylation process. We were looking at this reaction and studying it in detail and realized that pre-catalyst activation was foundational to how we think about being successful in homogeneous catalysis. And it actually came down in this case that just the order of addition of reagents and reactants was critical in controlling what active catalyst was formed from the same common set of reagents, the same pre-catalyst. So this palladium acetate, for example, if you treat that first with B2-Pin2, will generate a palladium-0 species that's bisligated with phosphine. However, if you treat it first with an aryl halide and base, you'll generate this monoligated palladium species as the active catalyst. And these two terminal outcomes could be mixed or mixtures, which impact reactivity significantly. And indeed, if you control the formation of these two distinct catalysts, you can look at their different rates and reactivities. This was a really important lesson for us to focus on this element of catalyst activation. It was reinforced actually by the second route that we looked at to the same molecule. It's all towards this JAK2 inhibitor. And we were looking at this CH arylation chemistry. And it was really a surprise to us that actually bidentate phosphines turned out to be best in this chemistry. But when we made the bidentate phosphine complex oxidative addition adduct, this turned out to be completely catalytically inactive. And this was great work we did with Yining and Donna Blackman at Scripps at the time. And it turned out that during the pre-catalyst activation, one of the phosphines on the ligand was getting oxidized as we made the palladium-0 catalyst. And this mono hemilabile bisphosphine was really, really critical to the success of the CH arylation chemistry. Now, why was this important? It was important because as we moved on into more complex spaces, so we get back to this BTK inhibitor again, and we looked at trying to make the axially chiral aryl-aryl bond, and we wanted to make this with the simplest ligand that we could possibly get away with. It turned out that BINAP would function, but only if we controlled the catalyst activation to specifically generate that hemilabile phosphine monoxide. And when we did that, BINAP could form this axially chiral bond with a fantastic diastereoselectivity selectivity and excellent yield, and gave us a simple way to start thinking about making that molecule. So why is it relevant? Well, as we think about approaching novelty and getting into new chemical spaces, bringing base metals or other technologies into our organization, this is the kind of mindset we have in the process chemistry team. So this community and group at the bottom here have really been working hard in these spaces to collaborate externally and to develop science internally that's going to support these priorities. Now, one of our first explorations has really been in nickel chemistry, to explore the use of nickel in our portfolio. And we've done this through innovation, through reaction design, but also internally, as I'll show you. It's important for us to explore the novelty of nickel, not just repeat palladium chemistry, but the new reactivity that we could leverage through this reaction manifold. So we've looked at things like nickel redox active ester chemistry with Phil Barron, or nickel difunctionalization chemistry with Curie. We've expanded into other metals such as copper with Marisa Kozlowski, and been doing a lot with Paul Chiric on cobalt chemistry, for example. So how do we think about developing these into active things that we can use in our portfolio? Well, that requires this kind of mechanistic insight in terms of how these catalysts are activating. So Steve Wisniewski and Matt Juano have been developing protocols with Eric Simmons and the rest of the team about how we even think about screening for reactivity in something like base metals. So taking, for example, this simple nickel catalyzed borylation chemistry, the team realized that phosphine ligands don't complex to nickel 2 salts in methanol like you would expect in palladium chemistry, and that can have some really negative consequences. So Matt discovered that, for example, if you take nickel dichloride in methanol and treat it with either a little bit of base or a little bit of methoxide, you'll quickly generate this nickel alkoxide polymer that completely precipitates from solution and becomes completely unrecoverable and catalytically inactive. And you can see some pictures of that polymer there. Now, as we think about scaling up nickel chemistry, this sensitivity to base and alcohol obviously becomes a major issue for large-scale applications. So Matt has done a fantastic job exploring the space and understanding how nickel 2 halide could be transitioned into active nickel 0 catalysts. So, for example, looking at diboron reagents again and their ability to reduce nickel dichloride, for example, nickel B-neo will do the reduction reasonably well. Diboronic acid will do the reduction, but things like B-cat won't. Nickel bromide will function as in the reduction, but nickel sulfate and nickel nitrate won't. This is really important knowledge for how and what is reducing these pre-catalyst structures, and this has allowed Matt to develop some really nice protocols for converting nickel 2 dichloride into nickel 0 catalyst structures, such as this complex phosphine or even COD complexes that Matt was able to pair on really great scale and with great selectivity. Now, Matt and the team have continued to explore this space computationally to understand in more detail how this process happens. And it turns out that once you expose nickel dichloride or nickel methanolate to things like Hunig's base, you'll get complexation events that occur. And those complexation events actually increase or decrease the pKa of the alkoxide or alcohol protons that are bound to the nickel species. Computationally, this is all downhill. As you bind more Hunig's base onto nickel, you end up with these bi-ligated species or these tri-coordinate nickel complexes that displace the alkoxide, and actually these are the ground states thermodynamically, but these are catalytically irrelevant pre-equilibrium. What needs to happen is a deprotonation event of the alcohol ligands to the alcoholate over on the right-hand side there. However, once that deprotonation event happens, this is how we get the polymerization into that nickel alkoxide polymer. So as this complex is treated with base and that deprotonation event happens, from a setup perspective, we need to make sure a really active reductant is present and a really active ligand is there to capture that activated metal species and drag it into the active catalytic cycle. And this understanding that Matt has developed, as highlighted in this organometallics paper, is foundationally important to how we think about continuing to develop base metal catalysis. With respect to catalysts, we've continued to focus on how we can bring innovation in the nickel space. So Van Tran, who was in Kiri Ingles' group, came and did an internship with us as part of our deep collaboration with Kiri's group. And during that internship in New Brunswick, we kind of stumbled across an old complex called nickel COD-DQ, which we found to be remarkably air stable, but also a functional pre-catalyst for catalyst activation. And this led us down into a really interesting space where we wondered if we could supplement different ligands onto nickel 0 precursors to enable smoother catalyst activation. And we developed a protocol with Kiri for not only screening for nickel complexation, but thinking about how to select different nickel complexes that might be functional and stable and how to study their properties, especially with respect to oxidation potential, to identify reactive air-stable nickel 0 precursors that we could use in our own portfolio. So through this great collaboration that's gone on for the last couple of years, we've developed, I think, 10 novel air-stable complexes at this place, at this standpoint, with 3 different structural types of class of new nickel 0 ligand, all of which have different activation properties and kinetics and different oxidation potentials. And we're just in the process of writing up this manuscript, which we'll be publishing on in due course. Now, that's not to say we're ignoring the unique reactivity of nickel. So that members of the team have been looking at how to encourage novel reactivity and explore that in our portfolio. So Greg Boynton has been doing a fantastic job exploring nickel chemistry. For one of the examples he's published on is this isocyanate insertion into this anhydride compound, which was of relevance to that compound I've showed you a few times now. So how we think about this innovation space and bringing new capabilities to our group really is a mixture of deep mechanistic understanding and innovation in application. Now, you may have noticed that we were focused not just on base metals, but also on photochemistry, electrochemistry, and flow. And the reason to do that was we thought that photochemistry and electrochemistry might teach us about single electron control elements, and that would help us understand how to manage the reactivity of base metals more. And if we're going to do any of those, we need to be good at flow chemistry. That's why those 4 topics are innovation areas for us at Bristol Myers Squibb. And we're just starting to get to the place now where we can really explore all of those different areas and their relevance into molecules in our own portfolio. So for example, this TLR789 agonist that was published on last year by Al Diekman and his team contained this really interesting sp2-sp3 bond, which we made and process scale through a pretty obvious route of taking the vinyl boronate, doing a standard cross-coupling, and then hydrogenation to make this sp2-sp3 center. But bringing nickel and these different elements together, we can now look at sp2-sp3 couplings through different manifolds, through kind of photo redox enabled nickel catalysis or nickel-based Suzukis or electrochemically mediated nickel chemistry, or even some of the cross-electrophile couplings, either with inorganic or organic kind of reductants, and explore which ones are easiest to optimize. How does optimization occur with these different manifolds? What are the differences in impurity profile, yield, and cost of reagents, reactants, etc.? What approach is going to give us the most sustainable outcome? So Greg's done a fantastic job with Candace, Steve, and Eric Simmons to look in this specific example how all of these different reaction types perform from those different parameters, yield, optimization, potential, etc. For example, the lowest yielding one here, or one of the lower yielding ones, is a cross-electrophile coupling with manganese, but the coupling reagents you use are so much easier to make. This might be the most optimal position to think about. So this JOC paper goes into a ton of detail about that activity. Simultaneously, we've been exploring how to scale up cross-electrophile couplings using manganese, for example, a standard Weiss-like coupling with our group at BBRC in India, which I'm very grateful to be able to work with on a regular basis, and see what scale-up parameters are important. For example, particle size of the manganese dust, how to get suspension to run these reactions on kilo scale, and they've published this great paper in OPRD on that topic. So that's how we're bringing these elements together. The BMS portfolio is not just for small molecules. We've got degraders, as I mentioned as well, but also millimolecules, these fantastic biomolecules, whether it's macrocyclic peptides or oligonucleotides and other kinds of innovative conjugates with biologics, antibody drug conjugates, for example, and other modalities that force us to think about chemistry and the challenges in this broader and different context. And this union between medchem, process chemistry, and academic collaborations has really been critical to us thinking in this space. The one I want to highlight is really the outcome of a remarkable team that's been pulled together from our colleagues in drug discovery, Rick, Ivar, Stephen, and Yaz, the process chemistry team, Mike Schmidt, Bin, Shenji, and a large number of folks in the process group, and the amazing team of Phil Barron to think about antisense oligonucleotide chemistry. So back in 2016, we were really interested internally in starting to get into this space, and we thought very much about antisense oligonucleotides, compounds like Spinraza, which are 5-phosphate-linked nucleotides. Okay, so in the polymer backbones are linked together by these 5-phosphates, which traditionally are made in an unselective fashion. So a mixture of stereoisomers is created, and for an AIMer like Spinraza, that might be up to 132,000 diastereomers. Internally, we'd worked for a while on these protide prodrugs, these chiral phosphate prodrugs and nucleosides, and developed some innovation on how to make those selectively. And we were inspired by that work, plus the work of some giants in the field. So specifically Wojciech Stek, who worked on these oxalate phosphorylane chemistries as a stereo-specific way to couple nucleophiles onto phosphates, for example. And we wondered if we could generate a reagent platform that would allow us to glue two nucleosides together through a chiral 5-phosphate linkage. That allowed us, or that thought process, the outcome was what became known as a Psi reagent, which stands for phosphorus sulfur incorporating reagent that's derived from limonene, and gave us a way to make chiral 5-phosphates in oligonucleotide chemistry and make therapeutic length oligonucleotides. And we were given graciously given the 2021 Green Chemistry Challenge Award by the EPA this year for that work, which we published in 2018. Now, what wasn't clear to anyone outside of BMS at the time was we were actually simultaneously to developing that chemistry with Phil, applying it into the BMS portfolio in a completely different context. So we're looking at the synthesis of these highly complex cyclic dinucleotides, which contain not a diphosphate linkage, but an azaphosphate analog in this highly sterically encumbered environment to make this very complex macrocycle, which we hoped had efficacy in the native immune system. And we had to develop a novel version of the reagent to act in this environment, but we could take these aza sugars, derivatives, and this new reagent and link them to make these macrocyclic cyclic dinucleotides with excellent diastereoselectivity, as you can see, over 200 and 100 to 1 for both of those phosphorus stereocenters. We published on this in JOC this year. And what was great about this work was it wasn't just the process chemistry team advancing this clinical candidate. James Kempson and the discovery team with Peter Park were using this chemistry to explore novel analogs simultaneously to us delivering on this innovation, which I thought was an outstanding way to connect that community together. Now, bringing these different minds from academics, industry together led to this reagent being thought of as applied into really creative spaces, which I don't think our team internally would have necessarily thought of. So Dong Min and Nazareth and Phil's group thought about adding carbon nucleophiles into the Psi reagent platform, and we were able to show that you could sequentially add Grignard and alkyl lithium and another Grignard into an analog of Psi to generate chiral at phosphorus phosphine ligands. So completely outside of where we were thinking, into intersecting a new field of chiral asymmetric catalysis. Once we've been able to add a Grignard, we were able to make neutral methyl-derived alkyl phosphates, and the team developed chemistry to link nucleosides together now, not with a phosphoric acid derivative, but a methyl-derived phosphine derivative. The team thought about the ability to use the Psi platform to do bioconjugation, and when they explored the chemistry of the Psi reagent platform with different amino acids, found that there was a great deal of selectivity for conjugation on serine. So we're able to develop a bioconjugation protocol for site-specific bioconjugation onto serine residues within peptides, for example. And collaborating with Phil Dawson's lab, we're also able to show we could do site-specific conjugation on oligonucleotides, and we were able to actually add on tags that allow us to do site-specific conjugation onto antibodies. So it gave us a way to generate antibody oligonucleotide conjugates in a site-specific fashion, which is a completely new area for us to think about in terms of biological activity and pharmaceutical properties. The last two slides, I'll leave you with the story about how we wanted to squeeze some more out of the lemon peel that we use, or orange peel that we use to make the Psi reagent. We wondered if we could apply it not just to the chiral thiophosphate chemistry, but to parent phosphate linkages or even dithiophosphate linkages, and generate a platform for making oligonucleotides that operated seamlessly at P5, at phosphorus 5 oxidation state, and would allow us to make chimeric structures where we could program in and modify at will the very backbone linkage phosphates chemistry and open up easily a new form of SAR for our discovery colleagues. And that took us a couple of years to work on, looking at the very sensitive reactivity of phosphorus 5 and very subtle reactivity of phosphorus 5 to modify what's on the ring system, what's on the leaving group to enable this to occur. But we developed a series of P5 reagents now that allow you to make a methyl phosphate, the chiral thiophosphate, the racemic thiophosphate, a dithiophosphate, all the parent phosphate linkages, and install them on a same coupling condition, homogeneous platform and oligonucleotide synthesis to make these chimeric molecules where we're able to vary the very backbone of the molecule itself. And you could make these chimeras with the same yield and purity that you could do with one of our standard oligonucleotides. And we're really excited to see some great coverage of this paper that came out just a couple of months ago or so. So what's the outcome here? What's the story that I'll leave you with? The conclusion is that bringing diverse mindsets together to think about these complex problems pushes science in really interesting directions. And it's been really cool to see the Psi reagent platform connect transition metal chemistry to small molecules, to oligonucleotide chemistry, to chimeric structures, to bioconjugation. And I don't think we would have unearthed this plethora of potential impact without the union of these different minds from medchem, process chemistry, and academics. And as we think about some of our more pressing challenges or new challenges in sustainable science, in late-stage functionalization, or how to think about more advances in tides, continuing to collaborate as an organization outside of just our own spheres is really, really, truly important. So I'll leave you there. Hopefully I've been able to highlight to you the main protagonists of every story. This is their story, it's not mine, but there are hundreds more people who have been involved in these projects than I could possibly acknowledge on one slide. So thank you to everyone who's touched the work I talked about today. Accelerating through discovery and development is really important to us. And if you're interested in joining our team to help patients, please reach out. We always have positions open in our organization for talented folks. So I look forward to hearing from you. And I'll leave you with the slide in case you missed any references. Scott, Carl, thank you so much for the invite. Rio, thanks for coordinating the meeting. I'll be more than happy to take any questions.
Yeah, wow, thank you, Mark. That was great. That was
Thanks,
great.
Mark.
Maybe I would love to start. I'd love to go back to the lab. That'd be great. It sounds like a really exciting place. It sounds like a great place to start your career, right? Like where you guys are really trying to be creative and curious and in an effort to make medicines and help people. So did we have any questions queued up? I had a few.
I don't see any queued up, but as well do I. So go ahead, Carl.
Yeah, I guess my first question, isn't it a chemistry related one? It's more of a data related one. So like in the beginning of the talk, you talked about all these different, what was it, like 20,000 experiments or something like that, that you'd leveraged on a number of different platforms in this one project, right? And I guess the question I have is like, how do you, like, you create a tremendous amount of data from that, from any one experiment, and then you multiply that. How are you guys thinking about how that data is set up and accessible, within your team, across BMS? I mean, it's like it's one of the really like real challenging problems, but I think like kind of really solvable that you've been coming up. So I'm just curious if you have any insights about that.
Yeah, it's actually a really important problem, I think, Carl, how to connect, especially in chemistry, where we use a lot of diverse tools and analytical techniques that don't always talk to each other very well. And how we amass that data set to make disruptive decisions, I think, is important. We've been able to curate a lot of our screening data onto a single database on a standard way of reporting out the data that allows us to amass our catalysis data really, really well. But there's still a heck of a lot of cleaning of that data needed to get to the place where you can actually model against it, right? We're starting to explore new places, the data lakes and other areas where we can deposit all of our data and have access to it. But I think there's still some important metadata connectivity that's going to be needed for us to truly leverage data in new ways. A lot of our current modeling approaches really take subsets of the data. There's a cleaning enterprise that the group has to go to before we can do a lot of modeling information. But being on the cloud, leveraging data lakes, how to metadata our data is really the questions that the team are really starting to get into in more detail. And I think we need a lot of support from a lot of the instrument providers as we think about connecting different approaches to molecules and to data acquisition. We really need support from that broader community to be able to answer that question, to have data seamlessly connected.
Yeah, I mean, and so most of it's kind of driven by the questions you're asking up front, right? Like, like what will be at this particular time on a chromatogram or whatever. But the reality is, what you really want to do is not have to ask a lot of questions up front, right? Like, and be able to just, like pull through the information and look at trends. So yeah, it sounds like you might need.
Eventually, I hope. Yeah, I mean, I hope eventually data's amassed and accumulated in such a great way through the lab infrastructure that the team will be able to easily ask questions of that data, right? So you don't have to kind of curate it in order to answer a question. You can ask a question directly of the data set, right? And use Python and R and other modeling approaches to interrogate it.
Very cool. Scott, you want to hit?
Martin, yeah, I have a question, but somebody else brought it up as well in the chat. Great talk, obviously. Lot about nickel, and it's been been a really hot area in the last 10, 15 years. What's your concern about using nickel in process?
Yeah, I know there's some divergent opinions about that. As we've interrogated it with our partners, we haven't really had too much of a concern with respect to handling an occupational exposure. I think that's relatively easy to navigate. In terms of being able to take the nickel out of the reaction in a safe way, we've developed some place using thiocarbamates and other technologies to be able to take the nickel out of the reaction stream. And we haven't seen any inhibition to be able to access nickel from a sourcing perspective. Obviously, nickel is isolated on massive ton scale. So I don't see a huge number of issues that we can't manage there, Scott, from a toxicity standpoint. Obviously, in organic chemistry, we use a lot of toxic reagents and reactants that have acute and systemic toxicities to them. It's really important from a sustainability standpoint that we understand that and frame that appropriately, and also put the controls in place for reagents that are toxic and have problems. I haven't heard a lot of issues around nickel, to be honest, that are disproportionate to any other reagent or reactant that we use.
Awesome. Thank you, Martin. And so right now, guys, I think we're going to have to switch over to Sophie. Martin, there are a few other questions popping up in the Q&A, and folks, if you can hang on, and we'll get Martin over there so he can respond.
Yeah, you're free to answer them if you have the time.
Yeah,
thanks again, Martin. That was great. Really good
wonderful stuff. Thank you so much. And so,
no worries. Great to be here.
Thank you. So Sophie, if you can go ahead and share your slides, and while you're getting that ready, I will give a quick introduction of next speaker. So our next speaker is Professor Sophie Rousseaux, who is currently at the University of Toronto. She received her Bachelor's of Science in Biopharmaceutical Sciences and Medicinal Chemistry from the University of Ottawa in 2007, and where she worked with Keith Fanyu on the direct derivation of pyridine N oxides and decarboxylated ketone aldol reactions. She remained at Ottawa in the Fanyu group for the start of her graduate studies working on palladium catalyzed reactions, and then moved to Cambridge to work with Steve Buckle at MIT to complete her PhD. She then worked in France and at Oxford for her postdocs. And during her time at Oxford, she was a junior research fellow at St. John's College. In 2015, Sophie returned to Canada and is now in the Department of Chemistry at the University of Toronto as an assistant professor. And as a Canada Research Chair since 2016, her group's research include organic synthesis, catalysis, organometallic chemistry, and supramolecular chemistry. And I've been following her papers for quite a while now, and there's some fantastic work that I'm really excited to hear some backstory on. So I think you guys are all really excited to hear this talk. So welcome, Sophie. Welcome to our symposium, and take it away.
Thank you so much, Scott, for that really kind introduction. Hopefully everybody can hear me okay, and that all technical issues are great. I want to start by thanking Scott, Steve, and Carl for the really kind invitation to be here this afternoon to share with you some of the chemistry my group has been working on, I guess, in the past 6 years at U of T. And thanks, Rio, also for all their kind of technical magic to make all of this work. Okay, so before I get started, I actually just want to start or get started with the chemistry slides. I want to start by highlighting. I'm just going to move some of my Zoom windows out of the way. Okay. I want to thank and highlight those who have done all the chemistry that I'm going to be sharing with you today. So their names are going to be highlighted on the bottom right-hand corner of slides, but I do really want to kind of highlight just to start the names of the students who've done all the hard work that I'm going to be sharing with you today. Okay, so my group focuses on the development of new synthetic methods. And when I started my independent career, I guess 6 years ago now, we kind of drew up this mock catalytic cycle, if you want, for thinking about reaction development. And so the way we would kind of pick some of the methods that we would be going after would be, for example, either through targeting new types of reactivities, so reaction mechanisms that we thought would be interesting, or specific targets that we thought would be of interest to end users in industry. And ideally, once we'd be able to kind of develop methods that would kind of meet those two criteria, we would study the mechanisms of those transformations, because we hope through mechanistic understanding that we could ultimately get to improved catalyst and subsequently improved reaction conditions and reaction design, which would feed back into essentially new reactivity and new target selection. And so this kind of, if you want, catalytic cycle, if you want, for reaction development has worked really well for my group so far. And the main areas of chemistry that we've been working in is the synthesis and functionalization of cyclopropanols, although I would argue that we're kind of getting into more small ring chemistry in general, so cyclopropanes and cyclobutanes. And the other big area of research in my group has been the synthesis of nitrile derivatives using non-toxic feedstocks to incorporate the nitrile functional group. And so I'm actually going to highlight a lot more of this chemistry in my talk today, but I am going to talk a little bit about some of our cyclopropanol functionalization chemistry. And I actually chose, I guess, maybe following Martin in today's, I guess, lineup of speakers, I chose to focus my talk on some of the areas of nickel catalyzed cross couplings that we've been working on with respect to these two topics. But first, a brief overview of our interest in cyclopropanol chemistry. And so I won't have time to talk about the chemistry that we've developed on the first half of this slide today, but we have been very active in looking at cyclopropanols as synthons to access homoenolates, an interesting chemistry that you can do with these homoenolate equivalents. And so, for example, we've reported a method to take cyclopropanols and directly convert them to cyclopropylamines, what looks like a formal substitution of the alcohol with an amine, but these reactions actually proceed through ring opening of the cyclopropane ring to reveal this metal homoenolate, which can condense with an amine, generating this aminium, and then subsequent ring closure gives you that trans cyclopropylamine product. We've also used cyclopropanols, or the homoenolates that they generate, as nucleophiles in nickel-catalyzed cross-couplings to essentially forge new sp3-sp3 carbon-carbon bonds through these nickel homoenolate intermediates. And as I mentioned, we've been thinking about using cyclopropanols and nickel catalyzed cross couplings, and in particular, thinking about using the CO bond in the cyclopropane ring as a handle for cross coupling chemistry. And this is some of the chemistry that I'll be sharing with you in the first little bit of my talk. Okay, so why are we interested in using cyclopropanols as synthons in nickel catalyzed cross couplings? Well, in particular, what we kind of like thinking about in my group is the complementarity of using cyclopropanols, unlike other cyclopropane synthons in cross-coupling chemistry, because you can access these starting materials really simply from simple esters through Kulinkovich chemistry. And so essentially this offers kind of complementarity, if you want, compared to say Simmons-Smith or other carbanoid chemistry to access cyclopropane derivatives. Now, Reggie Mills, who was the first PhD student to join my group, started looking at kind of developing a cross-coupling reaction where we'd functionalize this CO bond very early on in his PhD. And it became quite apparent relatively quickly that any 2-electron chemistry that we would attempt to do, so where oxidative addition to the CO bond would occur through either an SN2' or an SN1 type reaction, would lead to ring opening of the cyclopropane to generate this nickel pi allyl species. And so ultimately, if you tried to trap the nickel pi allyl species with the nucleophile, you'd get to these types of functionalized products, which can be interesting in themselves. But as I mentioned, we wanted to retain the cyclopropane ring in the products we generate because cyclopropanes are important motifs in pharmaceuticals, as we just saw in several examples in Martin's talk. And so what Reggie quickly realized is that to functionalize the CO bond and retain the cyclopropane ring, we would have to take advantage of the 1-electron chemistry available to us using nickel catalysis to essentially cleave the CO bond in a homolytic fashion. And so this would hopefully allow us to retain the cyclopropane ring and generate this, I guess, nickel intermediate, which can then go on to cross-couple to give us the functionalized cyclopropane product. And so if there's really one kind of take-home message from this slide, it is that there is a mechanistic dichotomy in terms of oxidative addition of cyclopropanol derivatives, and in particular, whether your leaving group on your cyclopropanol favors 2-electron versus 1-electron chemistry. And so the cross-coupling reaction that Reggie ended up developing is shown on this slide. It's a nickel-catalyzed Negishi cross-coupling using these aryl zinc reagents. And essentially, I'm going to pop up essentially a reaction heat map with a wide variety of leaving groups on the cyclopropanol starting material. But what I want to point out, as highlighted on the previous slide, is that there's 2 potential products from this reaction. This product 2A, which results from ring opening through 2-electron oxidative addition mechanisms, and product 3A, which was our desired product, which results from oxidative addition through 1-electron chemistry. And so as one would expect, leaving groups on the cyclopropanol derivative that you would think of typically using for 2-electron chemistry or polar oxidative additions, such as tosolates or mesolates, gave product 2A in very high yields. So you can see these yields of 96%. A wide variety of other leaving groups, including leaving groups that one would expect to work for CO functionalization, actually only gave trace yields of the desired product. And it's only really when Reggie started moving towards leaving groups that are reminiscent of Barton-McCombie deoxygenation reactions that he started seeing high yields of the desired product 3A. And you'll notice on the right, I've included a lot of different reaction conditions. This is, again, not to overwhelm with kind of specific details of the reaction. It's just to highlight that Reggie did screen a wide variety of reaction conditions for all of these leaving groups. And so the yields that I'm showing on this slide are really the reflection of kind of the yields under optimal conditions for each of these leaving groups. And so the yields that you're seeing directly reflect the inherent reactivity of the nature group, the leaving group, and not the reaction conditions that were used in this chemistry. Reggie, assisted by John, went on to explore some of the mechanistic details of this reaction. And I won't have time to highlight all of the work that they did, but I do want to kind of highlight our proposed catalytic cycle. One thing that Reggie and John found is that this reaction actually generates a thionoester intermediate as the first step in the catalytic cycle. So what happens first, we believe, is transmetallation from the aryl zinc to the nickel 1 species to generate this nickel 1 aryl intermediate. Single electron transfer to our cyclopropanol with the redox active leaving groups generates this radical anion here. Ultimately, fragmentation generates radical 5, which can recombine with the nickel 2 species to generate this nickel 3 intermediate. And from here, you get reductive elimination to generate, as I mentioned, this key thionoester intermediate 4. And depending on the electronic properties of this aromatic ring on the thionoester intermediate, it can also be redox active and accept an electron from a nickel 1 species to generate this radical anion, which undergoes that key fragmentation to generate the cyclopropyl radical. Recombination with nickel gives us that nickel 3 intermediate, and then ultimately reductive elimination gives us the desired product. And so what's really interesting here is that it's the second catalytic cycle that is really the product forming catalytic cycle in our process. I should point out that these starting materials, 1, are actually easier to access than the thionoester intermediates 4. And so that's why we've stuck with 1 as our starting materials in these reactions. And as I mentioned, the electronic properties of the aromatic ring here were actually important to get this second catalytic cycle to go. And so if Reggie and John isolated thionoester intermediates, if the thionoester intermediate with a paramethoxy group goes on to give the desired product in high yield, however, the thionoester intermediate with a parafluoro derivative actually only gives trace yields of desired product. And this actually was kind of surprising to us, considering that at this step of the catalytic cycle, the thionoester is actually accepting an electron. And so presumably the more electron deficient thionoester would actually be more prone to give us the desired product. And we were actually observing the opposite in our experiments. And so unable to really explain this using experimental chemistry, we turned to our colleagues down the hall. So my colleague, Alan Asperu-Guzik, and a very talented postdoctoral fellow in his group, Gabe Gomez, who looked at this using some DFT calculations. And so the step of the catalytic cycle that Gabe ended up looking at is shown at the bottom of this slide here. And so it's a fragmentation of the radical anion resulting from reduction of the thionoester intermediate to generate that key cyclopropyl radical. And so what Gabe found, the key kind of result that he found, is that the paramethoxy group behaves as a stereoelectronic chameleon. And I really like this term that kind of Gabe introduced to my group, where at the ground state, the paramethoxy group is acting as an electron donating group, as we learn in undergraduate chemistry. However, in the transition state, the paramethoxy group actually rotates out of conjugation with the aromatic system to align the CO sigma star with the pi system to actually accept some of that electron density. And what that does is actually essentially lower the energy of the transition state for CO bond scission, making it achievable under our reaction conditions. And in the system where we have the parafluoro derivative, we don't have that ground state stabilizing effect. And so overall, the transition state, we don't have that transition state stabilizing effect. And so overall, the transition state is higher in energy here, and the reactions don't occur. And so that's why we're seeing that the paramethoxy derivative can go on to give the desired product. So briefly highlighting the scope of this reaction, these are just really our first generation reaction conditions. And John, along with Katie, a new PhD student in my group, have actually been taking a lot of the mechanistic lessons that we've learned that I've just showed to you on the previous slides and are applying these to develop much milder and reaction conditions that are much broader in scope. And I hope to report those to you maybe at some point next year. But at least in our first generation reaction conditions, we were able to prepare a variety of functionalized cyclopropane derivatives, especially using our really efficient paramethoxy aryl zinc derivative. You'll notice that in all cases, we tend to need to have an sp2 hybridized substituent on the cyclopropane ring to get high yields. Aliphatic derivatives do work as well, although yields are slightly more modest here. And as I mentioned, John and Katie are working on solving that problem, and we do have a solution to that that we'll report in due course. If you move away from that kind of ideal paramethoxy aryl zinc reagent, other aryl zinc derivatives can work quite efficiently. And in cases where the aryl zinc derivative actually leads to a thionoester intermediate that is not redox active, you can actually start the reaction using this kind of thionoester starting material, and you can get moderate yields of these cross-coupled products using kind of the combination of, or essentially focusing only on that second part of the catalytic cycle. Okay, so switching gears, I'm going to share some of our chemistry on essentially new strategies to access nitrile-containing molecules that avoid the use of cyanide salts, is essentially it. So one area of research that we've been very active is transnitrilation reactions, where we essentially transfer a nitrile group from one carbon-bound source of nitrile to a second, essentially, carbon-bound source of nitrile. And so these reactions, at first, we were using stoichiometric organometallic reagents to essentially affect these transformations, where the organometallic reagent essentially does a 1,2 addition to the nitrile. And if you kind of design the nitrile-containing substrate effectively enough, you can get this CC bond cleavage to occur to transfer the nitrile functional group. More recently, we've been looking at doing this chemistry using catalytic nickel under reductive cross-coupling conditions, so using stoichiometric zinc for this chemistry. And these reactions actually proceed through the same types of intermediates, and that's some of the chemistry I'll share with you in the next couple of slides. Other areas of interest in my group has actually been the synthesis of alpha aryl nitriles. So either using kind of feedstock materials such as cyanoacetic acids and converting those into N-hydroxyphthalimide esters or redox active esters to access these alpha aryl nitriles. And more recently, and again, I'll share this at the end of my talk, thinking about taking malononitrile and being able to use that as a platform to build up complexity and then use or lose one of the nitrile functional groups and ultimately producing alpha aryl nitrile products using this strategy. And so why are we interested in accessing nitrile containing motifs? And as I mentioned, a lot of the chemistry in my group is kind of tailored to synthesizing products or accessing motifs that we think will be of use to end users in industry. And so as you can see from this slide, alpha aryl nitriles on the top here and benzonitrile derivatives are found in a broad variety of pharmaceutical agents. And so obviously, not only is the nitrile functional group directly found, but it can be used as a synthetic intermediate, obviously, to access a wide variety of other functional groups. And so thinking back on kind of my training in palladium catalyzed cross-coupling, one of the conventional approaches that, I would think of in methods to access benzonitrile derivatives is through metal catalyzed cross-couplings using nucleophilic cyanide salts for these transformations. So essentially starting with aryl halides or pseudohalides and accessing these benzonitrile products in this way. And so there's a variety of metals that can catalyze these reactions. A couple of challenges include the use of sometimes toxic reagents in this chemistry, but I would say this is probably arguably a solved challenge or partially solved challenge with the development of potassium ferrocyanate as a reagent for these reactions. But more interesting from my perspective as someone interested in catalysis is the fact that cyanide is an excellent ligand for transition metals. And so catalyst poisoning is often a challenge in this chemistry. And just as I was starting my independent career, there was this really interesting report that appeared in JAX by Reeves and coworkers at BI, where they faced this exact problem of catalyst poisoning. And so the workaround that they developed, which I thought was really elegant, is that they actually converted the aryl halide to an organometallic and added this organometallic to a malononitrile derivative to affect one of these transnitrilation reactions. And so essentially the organometallic adds to the nitrile, and then you get this retrothorpe fragmentation to take place. And so what we wondered is, could we essentially affect a catalytic version of this reaction? So starting with the aryl iodide, not having to prepare the organometallic reagent in situ, could we directly convert that aryl halide to a benzonitrile derivative using catalytic nickel under reductive cross-coupling conditions? And ideally, what we were really hoping is that we would be able to have maybe broader functional group compatibility by essentially taking advantage of reductive cross-coupling for this chemistry. And so I'm not going to kind of go over all the reaction optimization here. I'm just going to highlight the kind of optimal reaction conditions that Reggie ended up finding. The reaction conditions are quite reminiscent of a lot of reductive cross-coupling reactions. So we use a nickel-2 pre-catalyst, zinc dust as a reducing agent in DMA. The transnitrilating reagent is a reagent that we've been calling MPMN for methylphenyl malonitrile. And this is really key to get the chemistry to work, is using this reagent. And so just highlighting a few examples of scope entries or that kind of showcase some of the broad functional group compatibility of this chemistry. You can use, say, functional groups that maybe wouldn't be compatible with the Grignard or aryl lithium chemistry that the Reeves group had developed at BI. So for example, free alcohols, esters, and amides all work well. Electron-rich heterocycles work well as well. You can use azine type derivatives. However, I will point out that a ortho substituent is required in these cases, because if not, these derivatives are too strong of a ligand or too efficient of a ligand for the nickel catalyst and essentially displace the BIP and poison the reaction. I do want to kind of highlight these two entries at the top here, where you have an aryl chloride and aryl tosylate functional group handle. In these cases, we see absolutely no functionalization of the carbon chlorine or the carbon oxygen bond. And so they're inert under the reaction conditions. Our proposed catalytic cycle is shown here. I don't have time to show some of the stoichiometric chemistry that Reggie did while studying this reaction, but we do have kind of fairly strong evidence, I would say, for the first half of the catalytic cycle. So oxidative addition occurs from low valent nickel to generate this nickel 2 species, and this is actually a reversible process under our reaction conditions. From here, we get a reduction to the nickel 1 aryl intermediate, and it's this nickel 1 aryl species that is responsible for the migratory insertion across the nitrile of the MPMN reagent. From here, some kind of formal beta carbon elimination occurs to release the benzonitrile product, generating this kind of alpha cyanoanion. And from here, presumably transmetallation and subsequent reduction regenerates the nickel 0 catalyst. And so, as I mentioned, chlorides and tosylates were completely inert under our reaction condition. And this kind of led Reggie to postulate that either oxidative addition was a challenge with these types of reagents, or maybe the single electron transfer, this kind of reduction step in the catalytic cycle, was a challenge when you had a chloride or a tosylate handle on the nickel. And Reggie had kind of other hints that kind of indicated to him that this might be, that the reduction could be a challenge in this chemistry. And that being when he used actually a reduction and or oxidative addition. When he used nickel dichloride pre-catalysts for this reaction, and I'll point out that our optimal conditions use nickel dibromide pre-catalysts, the reason for that is that you get a little bit of the halide exchange that occurs under these reaction conditions. And he could always observe some of the aryl chloride side product, which was catalyst active. If he used stoichiometric amounts of nickel, he saw much higher yields of aryl chloride under those reaction conditions. And actually, the way to kind of avoid, or not necessarily avoid aryl chloride formation, but essentially make sure that the aryl chloride that was potentially produced could be consumed, was to add a stoichiometric amount of a soluble bromide source in these reactions. So if he added sodium bromide or tetrabutyl ammonium bromide under these reaction conditions, he saw essentially no recovery or no formation of the aryl chloride, and instead he got much higher yields of the desired benzonitrile product. And so he could take kind of the results from entry 3 and the insight that he kind of got from that reaction optimization, and actually looked at a reductive cyanation using aryl chloride starting materials. So again, these are our standard reaction conditions with no additive at all. You get absolutely none of the desired transnitrilated product. And when you use a soluble bromide additive, such as sodium bromide, you now get a complete turn on of reactivity and you get 53% yield of the desired product. You'll notice that there's been no reaction reoptimization in this case. It's literally just an on-off switch for reactivity. And presumably, if you really wanted to, you could probably optimize the reaction to get even higher yields with these aryl chlorides. And so this effect of adding soluble bromide salts to turn on reactivity is not just an effect that is exclusive to aryl chlorides. We actually see it with aryl triflates, tosylates, and mesylates. And we even see it in some cases with actually very sterically hindered aryl bromides. And so you'll see in these cases here, the yields in parentheses represent the yields in the absence of the sodium bromide additive. And again, there's been no reaction optimization. It's literally just adding sodium bromide. You get a turn on of reactivity, and you can get a modest to moderate kind of yields of the desired products under these reaction conditions. So we think that the role of the sodium bromide may be beyond that of just facilitating the reduction step in the catalytic cycle, as we see that when we have very sterically hindered aryl bromide starting materials, adding sodium bromide can actually enhance the yields of the transnitrated products, even though they are still quite modest. We do think that maybe the sodium bromide might be facilitating oxidative addition as well, maybe potentially through the formation of a nickelate intermediate. And so we've been able to use kind of the knowledge that we got through studying that part of the previous reaction to essentially extend this chemistry to preparing alkenyl nitrile derivatives. And so we can access these alkenyl nitriles now, starting from kind of alkenyl tosylates or triflates, which are readily prepared from the corresponding ketones. And you'll notice the reaction conditions are quite similar to the previous reaction conditions. We do have a slightly modified transnitrilating reagent. So we have this kind of methoxy MPMN derivative, or some that I've labeled A here. And what Josh has found is that he can prepare essentially a wide variety of alkenyl nitriles using alkenyl tosylates as starting materials. He finds that he needs to use a slight excess of tetrabutyl ammonium bromide to get these reactions to go. However, when you have more reactive alkenyl tosylates, so that are essentially adjacent to an aromatic ring, or if you use alkenyl triflates as starting materials, you don't necessarily need to add the soluble bromide salt to get these reactions to work well. And so this chemistry is not yet published, but we're hopefully wrapping up this story shortly, and you'll be able to see it in the literature soon. Okay, so in the last couple of minutes of my talk, I want to just kind of focus on the second half of the equation when it comes to transnitrylation chemistry. And so everything that I've shown you so far has been about taking either an organometallic species, as Reeves and coworkers had shown, or in our case, we're generating that organometallic species catalytically using nickel, adding it to a melanonitrile to essentially enable these transnitrilations to take place to generate kind of the desired nitrile product. And in all cases, I haven't really focused on it, but essentially we've generated this alpha metylated nitrile product as waste. But one could imagine a certain scenario where these R2 groups are actually quite valuable, and you've been able to quickly and rapidly install them using melanonitrile as a starting material, because the chemistry to functionalize melanonitrile is quite well established and sometimes can be quite straightforward. And so the question that we kind of asked ourselves is, what if R2 is more valuable than R1? Could we actually start developing chemistry where we would take advantage of these alpha metylated nitriles and not use them as waste, but instead using them as nucleophiles, for example, in nickel catalyzed cross couplings? And that would maybe allow us to generate some quite complex quaternary stereocenters. And so the chemistry that Reggie ended up looking at is shown on this slide here. We actually, or for the beginning of this kind of reaction optimization, Reggie started from the alpha metylated nitrile derivative, or the kind of to simplify the development of the reaction conditions. And so the cross coupling step in the process would be this one here. So from the alpha metylated nitrile, an aryl iodide as the electrophile, and essentially was looking at various reaction conditions to get a nickel catalyzed cross coupling to go. And so Reggie looked at this chemistry for quite a while, actually. And what he found is that he examined a wide variety of ligand classes, examined over 50 different ligands in those ligand classes. And the general trend that he seemed to see was that the ligands either seemed to shut down the reactivity, so you would see kind of very little reactivity at all, or have no effect compared to what we were calling a ligand-free reaction, but it's essentially a reaction free of exogenous ligand. And kind of looking into the literature, Reggie became very aware that benzonitrile additives have been found to be quite beneficial in nickel catalyzed cross couplings. And so we started kind of thinking about that a little bit more in the context of all of our reactions, where we had these various nitrile containing products and side products and starting materials. So as I mentioned, in the absence of any exogenous ligand, the chemistry does work. You get about 36% yield of the desired cross-coupled product 2A here, and essentially a 1-to-1 ratio with the product 3 here. And product 3, this anisole side product, actually is a direct reflection of a beta hydride elimination side reaction once you get transmetallation of the alpha anion to the nickel species. So essentially what we're looking at in this ratio here is the ratio of reductive elimination to form the quat center versus beta hydride elimination occurring at that step instead. And so obviously we want to favor reductive elimination over beta hydride elimination. Reggie found that by adding a little bit of benzonitrile to the chemistry, so about 20 mol%, he could get a boost in yield, although product selectivities kind of remain the same. And ultimately, by adding a gross excess of benzonitrile, he started to see quite kind of synthetically useful yields, and probably more importantly, a kind of really intriguing selectivity, an increase of the selectivity for reductive elimination over beta hydride, reductive elimination over beta hydride elimination. And so ultimately, using 10 equivalents of ligand or 10 equivalents of benzonitrile is probably not the most synthetically useful method. And it just kind of got Reggie thinking about, obviously there's been a big effect by increasing the concentration of benzonitrile. Could we kind of increase the effective molarity of benzonitrile by designing a ligand where the benzonitrile would be coordinated to the nickel in a bidentate fashion? And so Reggie looked at a lot of kind of, if you want, ligand modalities here, or kind of Lewis base coordinating groups to favor this type of interaction. And the kind of key ligand class that Reggie ended up finding is one where the benzonitrile has an orthopyrazole unit. And in these cases, if you use 12 mol% of the ligand, you start seeing product yields that are resembling the 10 equivalents of benzonitrile, and selectivities where reductive elimination is being favored over beta hydride elimination. He found that moving towards a paramethoxy, a derivative of this ligand, led to just a slight increase in the product yield and just more reproducible reaction conditions overall. And so this ended up being our ultimate ligand design. But a few kind of control, if you want, ligands or control experiments to really kind of support the idea of this bidentate coordination of the ligand to nickel involved preparing this ligand here with where the nitrile and the pyrazole unit are para to each other instead of ortho. So they presumably have similar electronic properties, but obviously can't coordinate in a bidentate fashion to the nickel. And what we see here is that product yields and selectivity start resembling that of just simple benzonitrile. And same for this ligand here, where we don't have the Lewis basic nitrogen of the pyrazole. So we instead just have a pyrrole derivative. In this case, we get much kind of product yields and selectivities that are starting again to look like just the benzonitrile ligand, or the reaction using benzonitrile as a ligand on its own. And so ultimately, as I mentioned, this ended up being our optimal ligand design. And so what the final kind of cross-coupling reaction looks like is shown here. So we can start with malonitrile as our starting material. You can use phenyl magnesium bromide as a Grignard reagent to affect that kind of decyanation process to generate the alpha metylated nitrile derivative. Although I should point out that methyl magnesium bromide also works for this step. And then you would generate acetonitrile as a side product in this reaction. So that's very much a possibility. You generate the alpha metylated anion, and then essentially that can participate in the nickel catalyzed cross-coupling to generate these types of products too here. And in all cases, essentially, we generally need to use electron-rich aryl iodides for this reaction. And so I'm showing selected examples of scope here. If you want to incorporate a more electron deficient arene in your product, generally speaking, it's better to have that in the original malonitrile starting material. So essentially, if this R1 group is electron deficient aromatic ring, that's how you would incorporate it in the desired product. And you can see that, generally speaking, products are obtained in moderate to good yields. And so with that, hopefully I've stuck to time well enough. Again, I want to thank the students that done all the hard work that I've shown today. I've tried to highlight their names on slides as I went along. Reggie Mills, I did mention his name several times, was the first PhD student to join my group, and I've highlighted a lot of key contributions to my research program in this talk today. I'd like to thank, obviously, everybody that we've been collaborating with over the past 6 years, those who've supported our research since we started in 2015. Thanks again for the invitation to be here today, and I'd be happy to answer any questions you may have.
Great. Thanks, Sophie. That was fantastic.
Well done.
Thank you.
In mission accomplished, I think there was 2 questions that you answered in the last part of your talk about the utility of the byproducts from the reaction. So I think one question I had was I thought it was really interesting. Well, I had a bunch of questions, but the one I wanted to ask you that came at the very end is you did a really nice job of taking advantage of effective molarity with that ligand, right? But like the selectivity never really reached the level of when you just sort of swamped it with benzonitrile. And so what kind of questions do you guys have about ligand design? I mean, there's just so many ways to kind of poke at it, but I'm sure you have some ideas or thoughts about how to go for more there.
Yeah, it's really tough because I have a slide on this, but in the interest of time, I didn't show it, and maybe that will make it easier because I've got a good catalytic cycle. So the challenge with this ligand is that when we did some mechanistic studies on the reaction, the turnover limiting step of the catalytic cycle is actually oxidative addition, which is surprising for nickel, but all the data that we have supports this. And that's why we need to use electron-rich aryl iodides for this chemistry. And so the challenge is that we have developed ligands that can give really good selectivities of, say, 5-to-1, where at this step of the catalytic cycle, we can favor the reductive elimination to get this product over beta hydride elimination. And one of the ways that we've done that is that actually if we put an electron deficient substituent here, and so you get tighter coordination essentially of the ligand to nickel. But the problem in those cases is that we get very kind of much lower yields. We're kind of stuck in the 20 to 40% yield range because we've essentially kind of shut down the first step of the catalytic cycle. So there's a really fine balance that seems to be occurring here to get this chemistry to go where we get good selectivities while also, while still allowing turnovers to take place. And so to answer your question, we have been able to get the selectivities of kind of 5-to-1 using certain ligand design, but then we get very low yield. So there seems to be obviously just having, 2 benzonitrile ligands on the nickel seem to be much more effective. And we haven't been able to reproduce that yet in a kind of ligand that we've designed in lab. But that's a great point.
Cool, thanks.
Yeah, I love cyanations. They're so useful. What a great, one of my favorites.
Cyano is the new fluoro, right?
You know what? It really is. Cyanos and sulfonyl fluorides. I was going to ask a question about the generality of the aryl iodide, but hoping that you could get into sp3s. But that gets me to the other question, I guess, is the R1, R2 there? I mean, can you do sp3s there, or is that is it really kind of down to?
So here?
Yeah.
I believe, if I'm not mistaken, we've always had an aryl group here. I would have to look back. I can't remember if that's because it doesn't work or if that's also just because of the ease of the synthesis of the starting material or just the way that Reggie went about making those starting materials. But yes, obviously, one of the areas that we're thinking about here is if we can have even more sp3 rich molecules being generated through this mechanism, for sure.
Yeah, because you can make a lot of neat little starting materials from malonitrile. Great little starting material.
Yeah, one strategy that we've done, and we haven't published this yet. I just need to, again, Reggie has completed all that chemistry before leaving the group, and I just need to find time to write this up. But essentially, we can decyanate the malononitriles, and here we don't need to have R1 equals aryl. We can have R1 and R2 being essentially more or less anything you want. And then instead of doing a cross coupling, we can do SNAR chemistry. And so that's another way that we've gotten around the fact that the aryl iodide has to be electron rich because of that slow oxidative addition. And then you can start getting electron deficient arenes on that way. And so that chemistry works really, really well.
Excellent, excellent. A couple other questions. I see one from in here that's related to this is can substituents from this is from Jake Weiner. Can substituents with steric centers introduced on L3 lead to enantioselectivity?
Yeah, that's a great question. So that's something that we want to look at a little bit more. I believe we did one kind of quick test as we were developing this chemistry, and the yields of those reactions were very low. I don't even remember if Reggie looked at it on the HPLC to see if we got any enantioselectivity, but that's in the works, where essentially you can imagine this being kind of a chiral unit and trying to get stereo control that way. But that's an excellent question. And
and one from earlier in the talk, I don't know exact, I think this was from, based on the timing, when you were talking about the zinc and nickel aryl cyclopropanes.
Yep.
Question from Douglas Tabor about what about orthomethoxy? You did a lot with paramethoxy for the star materials.
I'm trying to think. I feel like if I remember correctly, I can't remember if we've tried specifically orthomethoxy, but we have tried up a kind of almost like a souped up derivative where we've had, if you want, like 2, 4, 6 methoxys, where we'd have several methoxy units that could potentially have the same effect. And that chemistry hasn't worked. And that's presumably because when they're ortho, they just can't rotate out of plane as easily. But we've just tried that kind of on the one substrate, and I would have to go back to see if we've just done the single orthomethoxy derivative. But trying to add more kind of methoxys to have an additive effect hasn't worked.
Cool. I think that brings us to the end. I want thanks for Sophie for a great talk. Thanks to the questions, really good questions, and everybody attending. And I want to thank the CCHF and Rio and all the support we get from them to put this on, and Scott, especially Scott. He's like the man behind the scenes helping to pull all this together. He's in his control room in Groton. So thanks everyone, and we'll see you guys in November.
Yeah, thank you. Look forward to seeing you all again in November. Thank you very much. Well done, Sophie and Martin. Thank you.
Thank you very much.
Okay. All right. We're off camera now. Excellent.
Yeah. Thank you guys.
That was great. That was great. Yeah. Well done, everybody. Martin, great talk, bud. That was